Good morning, everybody. It is my honor to welcome you all to our first webinar for 2021 entitled COVID-19 Vaccine Explained. Thank you for choosing to spend your Saturday with us to learn more about this vaccine. I am Dr. Grace Perez-Lirio, former president and current board chair of the Philippine Medical Association of New England. I'm practicing physician, internal medicine, and primary care at the Beth Israel Deaconess Healthcare I forget the long name, Leahy Healthcare, Needham Heights. I'm filling in for our president, Dr. Candy Buencamino, who unfortunately is unable to moderate this morning's event. So just a few reminders before we begin. This webinar is being recorded. If you have any questions or comments, please type them on the chat function. We will try to answer as many questions at the end of the presentation. It is hard to believe that it was only a year ago when we identified the first case of COVID-19 in the US. At that time, we had limited to no understanding of the novel coronavirus. Fast forward to today, there are close to 97 million cases and over 2 million lives lost worldwide. While those numbers are astounding and frankly daunting, the COVID-19 vaccine has arrived, bringing hope and taking us closer to the end of this pandemic. As an organization, the Philippine Medical Association of New England is committed to provide this important educational resource to the community. When the young, the old, the healthy, the sick, the rich, and the poor are equally devastated by a vicious and unfamiliar virus, helplessness becomes dangerously familiar. Not only did the pandemic change the way we live and work, it disrupted families, schools, and communities that resulted in profound tragedies, personal pains, and economic hardships. In many unfortunate cases, it resulted in permanent and unrecoverable losses. The reality is we still do not know when or whether we will ever be able to resume life as we once knew it. The COVID vaccine is a welcome addition to responsible behaviors that we have all learned over the past year. It brings hope to ending this cruel and devastating pandemic. On that note, I'd like to introduce our special guest who will be speaking and giving us hope. And this morning, he is not new to us. He was one of our webinar speakers last year, which was the specialist series. Today, he will help us understand how vaccines are developed, how they work, its benefits and side effects, and more. He himself is actively engaged in COVID-19 research studies and clinical trials. He received his medical degree at the University of the Philippines and completed his internal medicine residency at the Roger Williams Medical Center in Providence, Rhode Island. He went on to pursue additional subspecialization in infectious diseases at the University of Pittsburgh. He is an NIH-funded scientist in the Division of Infectious Diseases working on translational and clinical HIV inflammation and cure research. He also serves as the Associate Director of the Pittsburgh Immunology Specialty Lab of the AIDS Clinical Trials Group. Aside from his research, he also has clinical responsibilities at the UPMC Presbyterian Montefiore University Hospitals, serving as a consultant. His laboratory is actively involved in processing specimens from COVID-19 patients and from participants of COVID-19 vaccine trials being conducted at the University of Pittsburgh. Please help me welcome our distinguished speaker and guest this morning, Dr. Bernard Macatangay. Thank you for joining us again.
Thanks, Grace. I'd like to thank the organizers for this invitation. Next slide. As Grace mentioned earlier, the COVID-19 has really, more than 97 million global cases of COVID-19. This is as of yesterday afternoon, and over 2 million have died from this infection. Nearly a fifth of this is from the United States alone. It has not only affected, of course, people who got infected with COVID-19, but really it affected the economy of different countries around the world. It has greatly affected, of course, the most vulnerable in the population. And the next slide. So in the next hour or so, this is going to be the outline of the presentation. I'll talk about the virus and the immune response to the virus, which is going to be very important in trying to understand how the vaccine works. I'll give an introduction into the different vaccine types and the vaccine development and how fast the vaccine development for SARS-CoV is, and this is really a testament to the passion of a lot of different scientists and governments in trying to find a way to control this pandemic. We'll talk about the current development of SARS-CoV-2 vaccines, particularly the two mRNA vaccines that are available in the United States right now. We're going to look into the clinical trial data and also a little bit on the two Chinese vaccines, the Sinovac and the Sinopharm, specifically the phase 1-2 studies for these vaccines. And then we'll go on to some frequently asked questions regarding COVID vaccines. Okay, so we'll start with the virus and the immune response. The next slide. Coronaviruses, they were first described in the 1960s as your etiologic agents of your common cold. These are single-stranded RNA viruses with spike glycoprotein. There have been 7 of these viruses that have been discovered. And although the first ones that were discovered just caused mild disease, since 2003, as you can see there in the table, there are 3 coronaviruses that have been shown to cause severe infections and even with significant mortality rate. The most recent, of course, is SARS-CoV-2. And it's through interspecies transmission that contribute to the emergence of new coronavirus strains. Next slide. So if you look at the virus, it is composed of your genomic RNA and a nucleocapsid protein that is inside this phospholipid bilayer that contains other proteins. It's important to know the structure of the virus because then it will have an understanding of what the antibody targets. So the current vaccines that we have right now target these 2 proteins. Mostly it's really the spike protein. And the spike protein is important because this is the protein that attaches to the host cell. It has the receptor binding domain here in the spike protein, allowing the virus to infect human cells. Also, nucleocapsid protein is there, but most of the vaccines really target the spike protein. On the next slide. So we look at the viral life cycle, and here, the viral life cycle starts with the binding of the virus to the host cell. The virus, once it's incorporated, then releases genetic material. That the genetic material is then translated into the structural proteins, and then they are released in the cell. What has been shown in the early studies of SARS-CoV-2, early parts of last year, when they looked at the viral dynamics, is that the shedding occurs, what they did is they looked at throat swabs from different patients, and they showed that shedding typically occurs about 2 to 3 days even before symptoms start. So even before you actually see patients having the common symptoms associated with COVID-19, viral shedding is already occurring, which means that they're able to transmit the virus. And then soon after the symptoms start, the viral shedding then declines. And if you can see there, there's a lot of variation regarding viral shedding, right? There's some individuals that have really, really high viral shedding early on in the disease, and then it slowly goes down, which is the typical that you see, that dark black line that's there. But there are some individuals that somehow in the middle of, day 7 to 8, they would still have high levels of virus. So even if typically you see a decrease in viral shedding, there are a lot of different cases, because some people would ask, oh, there's someone that got infected or had symptoms at this time. Yes, although this is the typical viral dynamics of the dynamics of viral shedding, there's a lot of variability as far as shedding between different individuals. Next slide. So this is the immune response. The viral life cycle, as I mentioned earlier, would bind to the host cell. And so what really happens, and this is something that's very important as far as vaccine development, right? We need to know what happens to the immune response. And here, what you can see is that the immune response is when the virus attaches to the host cell and starts producing its proteins. The innate immune response is the first thing. So it's the first line of defense. What the innate immune response does is it tries to prevent, control, or even eliminate infection. And the first cells that you see here, the ones that have these sort of like tentacles there, are the dendritic cells. These are your antigen presenting cells. So once the body is infected with COVID-19, you have that antigen presenting cell, which then detects it. It says, okay, there's this invader, there's this virus. So what it does is it ingests the virus, breaks it down into several parts, and then it migrates from that point to the lymph nodes, where you have some of your T cells that are there. And what this antigen presenting cell does is then it shows the virus or these viral particles, peptides, to the T cells and tells them, okay, this is it, this is invading, do your thing. And so then you have your T helper cells, which can either help your cytotoxic T cells, which can kill the virus, or it helps form your B cells, which in turn then forms your antibodies. So this is typically what happens to the innate immune response. Next slide. So the virus, typically it's innate initial, the goal really of the immune response is for there to be long-term immunity, right? So that when someone becomes infected and then you have your immune cells already familiar with that virus, then what we want to do is that the moment the virus is introduced again, or we get exposed to the virus, our immune system already knows that it's there, and then it suddenly would produce the immune responses necessary to control and prevent disease of the virus. As you can see in the left side, we still do not know how long innate immunity or adaptive immunity is. So when someone becomes infected with the virus, we cannot fully say, or we cannot for sure say how long that person will have antibodies or immune responses that will prevent that person from becoming infected again. This is similar with the vaccine, right? The virus, we know what we know about the virus, we've known in the past year or so. So we can't say that in 2, 3, or 4 years that if someone becomes infected, if someone receives the vaccine, there's still going to be long-term immunity. That is part of what we're doing right now, is trying to monitor what happens to individuals who become infected or individuals who receive the vaccine to see what happens long-term. Will these people get infected? A lot of the questions, is it going to be like the flu vaccine that every year we're going to get infected, or we're going to get the vaccine? We don't know. Or is it going to be a one-time deal? You get the vaccine and you're going to be protected for the rest of your life. So those are some of the things that we don't know regarding the vaccine. And those are the things that a lot of the scientists and researchers are trying to get data from participants in the vaccine trial, or the patients or individuals who've received the vaccine in the past few weeks. Okay, next slide. So some take-home points. In the first part, again, viral shedding occurs prior to symptom onset. The levels are really high in the first week of symptoms. And then adaptive immune response, this would be your T cell and your B cell responses, can target several virus proteins, but the neutralizing antibodies, the antibodies that specifically target and neutralize the virus, are targeting the spike in the nucleocapsid proteins. The goal of your vaccine strategies, which we're going to be discussing in the next few slides, is to be able to generate these neutralizing antibodies and also elicit these T cell immune responses in order to control or prevent someone from getting COVID-19 or prevent someone from getting severe disease from COVID-19. Next slide. So before we go into the different SARS-CoV-2 vaccine candidates, let's look at the different vaccine types, because a lot of the different studies that are ongoing in SARS-CoV-2 vaccine will employ some of these different vaccine types. Next slide, please. Okay, so the first is your weakened virus. So again, a review of the vaccines. What do vaccines do? The vaccines aim to expose the body to an antigen that will not cause disease, right? So instead of someone getting infected with SARS-CoV-2 and going through this entire symptom phase and sometimes even getting severe disease, what the vaccine does is it will give you a little bit of the antigen in order for our bodies to create a robust immune response. And so that when the person becomes exposed again to that particular antigen, we already have some protection. All right. So the different vaccine types, weakened virus. So the virus types, either you give a virus that's inactivated, which means it's not going to replicate and cause disease, or you have a virus that is rendered uninfectious, or it's or weakened, again, which will not make it cause disease. And you give this virus into the individual in order to have the immune response. So what are some examples? So your weakened virus, this would be your MMR, your measles, mumps, and rubella. These are live attenuated viruses. Smallpox also. So there are vaccines that have been developed before that use this particular type of vaccine to cause an immune response, to build an immune response. Inactivated virus, this would be your flu vaccine. The yearly IM flu vaccine that we get, this would be an inactivated virus. And this is the virus that is developed by Sinovac and Sinopharm, which is the virus rendered uninfectious using some chemicals or heat and allowing it to be injected and cause an immune response. Okay, next slide, please. This are the new kids in the block, right? The nucleic acid vaccines, the DNA vaccine or the RNA vaccine. These are very safe and easy to develop. That said, SARS-CoV-2 or this COVID-19 vaccine from Moderna and Pfizer are the first mRNA vaccines that have been approved, right? So these vaccines are being studied for a lot of different types of vaccines for different diseases, but the first one to be approved would be that of the SARS coronavirus 2. And what it does is it basically allows, rather than giving the whole virus, it gives only the RNA or the DNA or the genetic material that's enclosed in this lipid layer or lipid particle. It's injected into the individual. The mRNA vaccine does not go to the nucleus, because I've heard people asking, oh, is it going to integrate into my DNA? No, it doesn't. The mRNA goes into the cell. It doesn't go into the nucleus. It instructs the cell to make the proteins based on its genetic material. And these proteins are then released. And so again, as you see here, that little tentacle cell, the dendritic cell, will then see those peptides and then cause the immune response, or will allow for the body to have an immune response. The mRNA is then destroyed after it's given this instruction. So there's no way for it to suddenly go into the nucleus, integrate into the DNA of the humans, and then cause some or alter the genetic makeup of that particular cell. So the ones that we have right now, they go in, they don't go to the nucleus, they instruct the cell to make the proteins, and then they get destroyed. And then these proteins are the ones that allow for the body to have this boosted immune response. Okay, next. Viral vectors. Again, so this is another that's been used in a lot of different studies. Like the Ebola vaccine has made use of viral vectors. There are clinical trials for prevention of HIV that use some of this, and also for therapeutic HIV that use viral vectors. The one that we have right now is the AstraZeneca vaccine for SARS-CoV-2 uses a viral vector. So what this does is that it, instead of giving the virus or SARS-CoV-2, is that it uses another virus that has been genetically engineered. This other virus does not cause disease. It's weakened, it does not produce disease. But what it does is it's genetically engineered so that it would produce the proteins of SARS coronavirus, as you can see here. So that when it gets into the cell, once it's injected, then the proteins are going to be produced by the body. But the virus itself will not replicate, and it will not cause disease. So again, it can either be, they use the adenovirus sometimes, or measles virus as the viral vector. But again, you might hear, oh, it's measles virus. It's not going to be, but no, these are inactivated already and weakened. They cannot produce disease. What it does is that it just brings the genetic material of SARS-CoV-2 into the individual. The AstraZeneca uses an adenovirus, chimp adenovirus. Next slide. And then these last vaccine types are either protein-based. So the first one, they give the whole virus. The other one, they just give the genetic material, right? The third one, they use another virus and make sure that the other virus has the genetic material of the virus that can then be injected. Here, they just give the protein, right? So rather than dealing with the whole virus or the genetic material, they give the protein. So for example, your hepatitis B vaccine is a protein-based virus. And so they use protein subunits, or either here, they also have viral-like particles where it's basically an empty shell. So it doesn't have anything inside it, but it just recreates the shell of the virus that has, the spike protein is there, right? So that when it's injected, then the body would see it as, an antigen, and then we'll have an immune response. And it's your HPV vaccine that uses this viral-like particle. So as far as protein-based, there have already been vaccines that have employed this type of vaccine type to create immune responses. Next slide. So a lot of people are talking about how fast everything went for this vaccine. This is the typical vaccine development pathway. As you can see, first, if there's a new virus or a new microbe that's there that's causing disease and we want to create a vaccine, it usually takes 3 to 8 years just to find out what we want to target, right? And then manufacturing, and then clinical assay optimization. It takes a long time. After this 3 to 8 year period, then you start with the first studies in humans. So this 3 to 8 year would include animal studies as well, or this is the preclinical phase. Then you start with a phase 1 study, which you look for safety, right? Is it safe? Phase 2, you have safety, immunogenicity. Sometimes you also look at the dose that you do. And then you have the phase 3, where you have thousands of participants would look at safety, efficacy. And this is actually the data that is submitted to regulatory commissions, or it's like the FDA, so that it can be approved. The question is, how were we able to come up with a vaccine in a year, right? When the typical vaccine development takes 8, 10, 18 years? Why? How were we able to do this? Part of it is because we knew about SARS coronavirus 1. So in 2000, 2003, when we had the SARS coronavirus, I don't know if people still remember that. I think finished medical school during that time, and there was this big, it was in the news all the time, SARS coronavirus 1. They were able to sequence that virus. They found out in SARS coronavirus 1 that the spike protein is important because neutralizing antibodies target the spike protein. So for any other microbe, that decision or that discovery to find out which the neutralizing antibodies would target will take years. But because we know SARS coronavirus 1, and they said, well, maybe it's the same thing. So the Chinese, they sequenced the virus by January, right? This started happening in 2019, end of 2019. By January of 2020, they sequenced the virus, and then they said, okay, it has the spike protein. Let's see if it works. And of course, the studies during that time showed that the neutralizing antibodies target the spike protein. So instead of the 8 years, it took us months to find out that this is it. This is where we want to do. This is where we want to concentrate to do the vaccine. So immediately you cut off up to 8 years of research based on that alone. So it's not like everything was just hurried and then people were not careful. No. This science, and being in an academic institution where we actually see all of this happening, you really see how careful everyone was in trying to come up with this vaccine. And then you have the phase 1, phase 2 safety. The mRNA that we have right now for Pfizer and Moderna, as I mentioned, this has been developed previously for other diseases. So all they needed to do was make use of what they've been developing in other diseases and make that sort of like the delivery or the vaccine type to come up with a vaccine. So I think, yes, there's a pandemic, but because of research that has been done previously, we were in a way fortunate that happened. So all we needed to do was to bring everything together. Also, knowing the process for getting grants in the NIH, let me tell you that they were reviewing grant submissions like on a weekly basis. So typically when you submit grants to the NIH, they're like 3 times a year where you can submit it depending on which institute you want to do. And then there's a study section, there's a group of people that will review it, and then it'll take a few months later before it's going to be approved, and then you get the funding to do the research. They were doing this research on a weekly basis. They were doing this review on a weekly basis. There's like a group of people that kept on reviewing these grants. So really, they made sure that all of the science and all of these information that we know right now, specifically in coming up with a vaccine, is done very quickly. But at the same time, there's a review process. So it's not haphazardly done. There's a group of people to make sure that the science being put forth there is really valid and really good science. Next slide, please. And so if you look at some of the examples of the viruses that had vaccines, look at the time it took for them to be able to develop a vaccine, several years. But then if you look at SARS-CoV-2, really within 12 months, right? So this started Operation Warp Speed. The goal of this was to have a vaccine by January 2021. What happened was the first, in the US, the first person to receive that was in December 14 of 2020. And this was an ICU nurse in New York. She was the first American to receive the vaccine. So even earlier than but the January 21 is for the distribution. And so by that time, I received my vaccine right before Christmas. So even before January, there was already the distribution of the vaccines. And a lot of people, specifically healthcare workers and people are are at high risk for getting severe disease, were getting some of the vaccines already. Okay, next slide. Take home points. There are several types of vaccines that are being evaluated for SARS-CoV-1. You have the virus, nucleic acid, viral vectors, and protein-based vaccine. Typically, the average time for vaccine approval is around 10.7 years, but this has been accelerated by Operation Warp Speed. And as I mentioned, the first vaccine was administered to Sandra Lindsay, who is a critical care nurse and the director of patient services at the Long Island, the Jewish Medical Center. Next slide, please. Okay, now let's look into the SARS coronavirus vaccines. So if you look at this figure, you see the different types that I mentioned earlier: protein-based, viral vector, DNA, RNA, all of that. And the numbers that you see there in that sort of like pie chart there are the number of vaccines that are being studied in each. So there are around 20 mRNA vaccine or RNA vaccines that are being studied. There are 3 live attenuated viruses. Inactivated is also there. So these are, even if we already have the 2 that's approved, so people are asking, why are we doing this, right? There are 2 approved vaccines. This is important, right? Because we don't know what the long-term effects or the long-term immunity that is caused by these vaccines. So the more vaccines that are available that we can study, the better, because some of them may actually have long-term immunity. Some of them may be able to work on viral variants that you have probably been reading in the news in the past few weeks or so. And so the more options that we have and the more vaccines that we can study that would provide really good, safety profile, but at the same time, highly immunogenic and very good protection against vaccines, the better. So the more, the better in terms of vaccine candidates. Next slide. So here you have, if you remember a few slides back, I showed you what happens preclinically. You have your animal studies, you have all of these manufacturing issues and whatnot. There are around 86 vaccines that are in that phase, which means they're not yet in human clinical trials. And then you have your phase 1 studies. Again, these are small studies. Safety is the main thing. Safety and what dose should be given. Phase 2 would be immunogenicity. So you have a little bit more participants that are enrolled in these studies. And then you have these large scale efficacy tests, which are your phase 3 trials. So there's 16 that are on phase 3 trials, and there are 2 that have been approved by the FDA. And this would be your Moderna and your Pfizer vaccines. Below you would see there a table of some of the leading vaccines. So you have, and of course, what the status of these vaccines are. So Pfizer, it's been approved in certain countries. It's already approved for emergency use here in the US. I received the Pfizer vaccine. Others have received the Moderna. That depends on state to state who gets to receive what type of vaccine, I'm sorry. And then you have the Chinese vaccines there. You have your Sinopharm, Sinovac. These are all inactivated. So again, similar to the flu vaccine that you get. And then you have your viral vectors, which is your AstraZeneca, the Oxford vaccine. That is in its phase 3 studies. We are participating in that study. And so we have been given either placebo or the vaccine for our study participants. And then you have the J&J also, that's ongoing study. So these are a number of the vaccines that are currently in development in some phase 3 studies and are also already starting to be approved in different countries. There will be a question and answer portion at the end. So just if you have any questions after the frequently answered questions, just list them in the chat box, and then we'll try to address those questions later. Okay, let's look at the clinical trial data. So I pretty much concentrate on those that have phase 3 data available, right? So there are 2 that were just published late last month, which are the Pfizer and the Moderna studies, both published in the New England Journal of Medicine. So the Moderna study, their mRNA 1273, is a phase 3 randomized, observer-blinded, placebo-controlled trial conducted in 99 centers around. So you're not talking about participant number of 100, 200. No, you're talking about thousands and thousands of participants. And the AstraZeneca that we're doing right now is like 30,000 participants. So the idea for these large phase 3 studies is to get as many data available for safety, right? Because if you have a small, phase 1, phase 1, you actually look at safety, but really things are different when it's already distributed to a huge population. And that's where you want to see what happens with safety. And that's what you want to see, where you want to look at when you want to see how effective it is in preventing, severe disease or the disease itself of SARS coronavirus. So here, there are local effects that have been seen, or local adverse events that have been seen in the Moderna: pain, erythema, swelling, lymphadenopathy. So these are the area where you get the injection. You would usually get some pain there. From personal experience, it lasts around 2 days. It was painful, I have to say. When I got my first dose, it was painful. After 2 days, it subsided. And you see very little grade 3 there, but mostly just mild and grade 1, grade 2 events. Next slide. Then you have your systemic events. Now, some people actually experience this after the first vaccine. Others experience this after the second dose of the vaccine. And it's classic fever, headaches, and fatigue, myalgias, or muscle pains, joint pains. Some may have some chills with it. I know of a colleague who, not the Moderna, got the Pfizer vaccine that really got like shaking chills or rigors. But these are moderate to severe side effects. Were noted about 50% of the vaccine group. And this is something that you would expect usually in a vaccine. I work on research looking at therapeutic vaccines for HIV. And sometimes when, we have participants and we give the vaccine and they say, oh, I got a fever, or I got chills. Part of it is like, that's good, because that means your body's actually mounting an immune response. In fact, when I got my second vaccine, I got a headache for 2 days, but I felt bad. I was like, I wanted to have chills and fever. I wanted to actually, and I felt like, I guess I'm older now, and my immune response isn't as good, because some of the younger colleagues, they were like, oh, I was getting chills. And I'm like, man, I didn't get it. So somehow I was like kind of disappointed with not getting side effects. Most of the time you don't want to get side effects, but some of these more systems like fatigue, myalgia, headache, this is usually really attributed to the virus, because then your body is having this immune response to it. But again, most of these, a great majority of these are transient, which means after about the second day, these symptoms disappear and they don't have sequelae. So it's not like you have this and then you will have some, symptoms that are long lasting. Most of the participants would have very transient symptoms. Next slide, please. And this is really what takes the cake, right? Look at the efficacy. So the gray is the placebo. The number of people who received placebo who developed the disease compared to those who received the mRNA. So this is the type of thing, and you see this and you're like, we're done, right? Like, look at the protection that is being given. And so this is 94.1% vaccine efficacy of the Moderna vaccine. And so when the FDA saw this, they're like, okay, we need to make sure that this is approved for emergency use. And so you could really see here that it was safe. Yes, you get side effects, right? You get adverse events, you get local and systemic. But if you look at the symptoms here, now the question is, but there are some that may, yes. So no one vaccine is really 100% safe. There will always be some cases where there may be an anaphylactic reaction, or there may be a very severe side effect. And this is the first time we're getting this vaccine. And so there are databases or registries for people who get these really severe reactions that they're submitted, so that people are able to learn more about it. We're on the same page. This is the first time we're seeing this. And so long-term effects, we still do not know. But the pandemic has really affected a lot of lives. And so you see something like this on a phase 3, you go, okay, this is good. Next slide. And here in this table, you actually see how it's different or divided depending on certain subgroups. For 18 to 65, you could see 95.6 vaccine efficacy, a little lower in people who are older. The immune response isn't as robust. And this is also probably the reason why you actually see more adverse events in people receiving the vaccine in the younger population compared to those who are older. They divide it there, male and female pretty much the same. We're trying to get in all of these studies, we're trying to get underrepresented minorities to participate in the studies, especially since there's some of the underrepresented minorities are really affected by this pandemic, this virus, this disease caused by SARS-CoV-2. We want to make sure that they are highly represented also in these vaccine studies to see whether the efficacy is going to be similar to Caucasians. Next slide. So that's the Moderna. This is the Pfizer. Again, if you look at there, so it's divided. The right and the left panel is divided into people who got it from 16 to 55 and those who are over than 55. Again, you see compared to placebo, there's a lot of reaction. So this is the vaccine that I got, and true enough, I did get the local effects of the vaccine. As you can see there, it's mostly mild and moderate. Yeah, maybe a little bit of a sliver there of some severe disease, but really it's really the green and blue, which are mild, moderate cases of local effects of the vaccine. In the next slide, you would see the systemic, if I'm not mistaken. Yeah, so these are the systemic effects of vaccine: fever, fatigue. Again, very much similar to the Moderna vaccine. As you see there. And again, you see more of these adverse events in people who receive the vaccine compared to placebo, which was expected. Okay, next slide. Next slide. And similar to that, so right, very much similar to Moderna. In here, so you get the 2 vaccine doses. Here, you see sort of like they magnified that first part. So after, these are days following the first dose of the vaccine. And if you can see where the red and the blue lines start to separate is around maybe 10, 11 days. So 10, 11 days from the time you get the vaccine dose, that's where you actually see the difference. And then if this is Pfizer, so we got our second dose at day 21, and you received by day 21, it continued to separate. And you could really see the difference between those who received the vaccine and the placebo, where they're really separate. And this is also 90+% efficacy of the vaccine. People are asking, well, actually look at after the first vaccine dose, you already have some improvement. Can we just give the first vaccine dose and not the second? The short answer is no, right? Because these studies, the analysis of these studies were done, or that 90% efficacy plus efficacy was based on the second vaccine dose, not the first vaccine dose. So we don't know what the effect is if we just concentrated on that. So all of the analyses, the primary endpoints concentrated on what the effect of the vaccine is after the second vaccine dose, which means we have no data whatsoever how protected someone's going to be just by receiving the first vaccine dose. Next slide, please. So this is similar to the table for the Moderna, where they divided it based on age. As you see there, I know you see, oh, look, this is 100% for some and whatnot, but also look at the number of individuals that were there who received it. They divided it also based on race or ethnic group and in certain countries. Because people say, oh, greater than 75, they're 100% vaccine efficacy, but the N is 700 plus compared to the total, which is over 27,000 and whatnot. So, 40,000. So, okay, so next slide. There's no data yet that I could find on phase 3 for the Sinovac and the Sinopharm. What I'm presenting here are the phase 2. So phase 1 and phase 2, again, phase 1, safety, phase 2, some of the immunogenicity or efficacy of the virus. But look at the N, or the number of participants. Phase 1 enrolled 144 participants. Phase 2 enrolled 600 participants, right? So this is compared to phase 3 of Moderna and Pfizer, which are like 30,000 plus. So you could already see the difference in that. That said, if you look at this, this is again a well-designed study, randomized, double-blind, placebo-controlled in healthy adults 18 to 59. So the reason why you see different doses and different intervals is because this is a phase 1/2 study. They're trying to find which is the most appropriate or the best dose, right, or interval of giving the doses. So it's either you get the 3 micrograms or the 6 micrograms separated by 2 weeks or separated by 4 weeks, right? Whereas in the phase 3, they already know what the dose is. They already know when to give the second dose. And so they were able to do that for 30,000 participants. And here you could see they separated the side effects or the adverse events based on the 3, 6, or the placebo. Again, similar to what we've seen, you would see the both systemic and local effects seen in those who received the vaccine. Again, this is different from the other two. The other two were Moderna and Pfizer are mRNA viruses. This is inactivated. So this is the actual SARS coronavirus that has been inactivated. So it doesn't cause disease, but once it's injected, it will generate an immune response. Next slide. So this is, again, this just the difference, right? In phase 3 big trials, you actually look at the number of people who got the disease compared to those who did not get the disease in the placebo and the mRNA. Here, they don't have that. What they show here are the neutralizing antibodies. So these individuals that were enrolled into the study, they were given the vaccine, and then several days later after getting the vaccine, they got their blood and looked at whether these individuals developed antibodies. And I'm talking not just of any antibodies, but I'm talking about neutralizing antibodies, or the antibodies that can target the virus and can neutralize the virus. And as you can see here, based on the 2 doses, 3 and 6 micrograms, that they were able to develop it compared to placebo, right? So placebo did not develop any neutralizing antibodies. But if you look at those who received the vaccine, they did develop neutralizing antibodies. Whether this leads to protection, again, at the end of the phase 2, you're not sure yet. It's only in the phase 3 where you actually see some of the results happening. And in the interim phase 3 trials, and this was, I don't know where this is published, they report 79% efficacy in preventing COVID-19. So it's interim phase 3. They don't have the full data yet for the phase 3 trials. Hopefully that's coming soon. And so we're waiting to see what it is. Usually when you get these phase 3 trials, even some of these interim phase 3, they can actually submit for regulatory boards so that they can be reviewed to see whether it can be already approved for emergency use. So in the US, we haven't gotten it yet. I think AstraZeneca is already trying to do that here. This is the Sinopharm, sorry, that's not Farm, phase 1/2 trial. Again, look at the number of participants, right? So this 96 participants who received either 2.5, 5, or 10 microgram dose versus the control. They received either 3 injections, and then you have the different intervals, either day 0 or 14 or 0 and 21. If you look at the adverse events, again, very similar to what we see in Moderna and Pfizer, where when you actually see that there's a local reaction that's there, or there may be more systemic reactions. The most common really is the local reaction. A lot of the colleagues that received some of these vaccines, not the Sinovac or the Sinopharm, did say that most of the time they did not get any of the systemic reactions, but all of them got the local reaction. Next slide, please. Again, similar to the Sinovac, they do not show us how many participants got the virus or the disease, but here what they showed are the neutralizing antibodies. So within 14 days or 21 days, you actually see high titers of neutralizing antibodies to SARS coronavirus 2. Okay. All right, so what are the take-home points? We have available phase 3 clinical trial results in 2 studies, and they showed acceptable safety and tolerability, and both of them would have local and systemic side effects that are transient. The studies show that the typical time where you experience these effects are around 15 hours from the time you get your vaccine, and then the classic time for you to stop that these symptoms resolve would be 2 days after that. Both the mRNA vaccines that are available right now in the US have greater than 90% efficacy after the second dose. Again, the data is for the second dose, not after the first dose. There are phase 1-2 studies from Sinovac and Sinopharm and some interim phase 3 data which showed safety and ability to generate neutralizing antibodies. How this would translate into whether participants or individuals who receive the vaccine are protected by the vaccine would depend on the final phase 3 trial results. And next slide. Okay, now let's go to some FAQs. Sorry, let me just get
Paige, actually, I think we have a very engaged audience, and we're going to ask we're going to call out there. I'm going to read the questions first. We so far have 5 questions. The first 3 questions are from Red from the Manila Times in the Philippines.
Oh, okay.
He asked, what do you think will be the effect of these vaccines on the new variants that had been spreading?
Excellent. So excellent question, right? That's all over the news right now. So there are several variants. There's a UK variant, which is the B117. And then there is also the South African one, which is, I think, the 501YV2. First thing, variants. Why are there viral variants? This really happens, right? Because the moment the virus replicates, there are mutations that happen. And so we expect these things to happen. What is important? So remember the first few slides, the antibodies, the neutralizing antibodies target some important proteins. So what you want to look for in these viral variants is whether the mutations that make it different from the original strain affects the protein where the antibodies target, right? So in this case, it's the spike protein. So what they found out in the UK strain, the B117, is that there were 23 mutations in that virus, and 8 of those mutations were in the spike protein, right? Including one that is in the receptor binder. Pfizer did a study to show that that mutation in the receptor binder, even if there was a mutation there, the neutralizing antibody was able to, well, neutralize the virus. The issue is they only looked at that. There are 23 different mutations, so we do not know. At this point, all of these studies are being conducted to see whether someone who gets infected with this viral variant will be protected by, well, if the person received the vaccine, then we'll see whether there is protection from severe disease. Or remember, there are studies that are also giving antibodies to people who develop COVID-19. So there may be some of these viral variants to see whether it escapes it. So the answer to the question is, yes, there are mutations that are there. We do not know for sure yet what these viral variants, what the implications of these variants are regarding to the efficacy of the vaccine.
Thank you. The next question from Red is on, can people choose their own vaccines? Should people be given a choice in their vaccines? I think there's a twofold answer to this question.
For right now, there are issues with the distribution of the vaccine and the availability of the vaccine. So at this point, at least here in the US, we don't really have a choice. we get the ones that are available that we get. Now, should people have the option? I mean, when we have multiple vaccines available, vaccine types that are available, and depending on what the side effect profile is and what the efficacy is, if they have been approved by regulatory boards, the FDA has approved them for use, and they're not only emergency use, but they're already approved for use, then I think it would be important for everyone to discuss it with their primary care doctors, see which would be the best vaccine for them. But I think having options would be very good, right? That's why we do all these studies, to see which one it is. And as long as it passes through the rigorous screening and evaluation of regulatory boards, right? We can't just, oh, this is available. If it hasn't passed through like the FDA and all of these things, we don't know what those side effects could be, right? And the main reason for these regulatory boards is to make sure that people who receive the vaccine are protected from what other events that can happen. And so, yes, I think if they're approved by the regulatory board and there are multiple people that have multiple vaccines that are available with acceptable safety and efficacy, I think we should have the choice to do that. At this point, though, we don't have it yet because there are issues with vaccine supply.
Yeah, thank you. Also, to add to that, here in the US, the states are regulating the distribution, even the criteria that will allow people to get it. And so while it is state regulated, we unfortunately will not have a choice. When we have
And
more supply of the vaccine,
yeah,
I think that will become that will change. But at this time,
one addition to that, like for example, here in Pennsylvania, the healthcare workers who were in hospitals, they received the Pfizer vaccine. People who are in nursing homes received the Moderna, and one of the main reasons for that is because the Pfizer vaccine, you need to have a -80 freezer to store all of these things compared to the Moderna. And so in big institutions or healthcare institutions, we do have a number of those freezers just to store the vaccine. So really it depends on the capabilities of the sites that are administering the vaccine.
thank you. And a third question from Red from Manila Times in the Philippines. Are vaccines advised to be used on those really frail individuals? How are the side effects of the vaccine affect those who had been really frail or have comorbidities? I
But
guess
yeah,
this is
so
asking, yeah, if you have comorbidities, should you get it if you're very ill?
given that there are comorbidities that predispose you to more severe disease, we really want, and in fact here, some of those individuals, like the elderly individuals, are now included in the 1A or the first tier here in Pennsylvania, because of what the virus can do to them, right? So it's either you get the virus and you get severe disease and could die from getting infected, or the benefit of getting a vaccine, where you saw from those phase 3 graphs, where you see really good protection, and that is from severe disease too, right? So if you look at the papers in New England Journal, they were able to prevent severe disease in individuals receiving the vaccine. So some of them may actually get it, but they did not develop severe disease. So for elderly individuals with comorbidities and frail who are at high risk for mortality from COVID-19, yes, it would be advisable for them to receive the vaccine. There are going to be side effects, but this is the time where we actually try to find out more to see how safe these vaccines are as they are distributed all over.
Thank you. Actually, to add to that, here in Massachusetts, we are about to vaccinate people who are 75 years old and above, and those who are with solid organ transplant or immunocompromised people. So it's actually, those are the people that we actually want to vaccinate. But thank you for that. Those are very good questions, Red.
One other thing that to consider is that, like for example, the solid organ transplants and stuff, remember their immune response may not be as robust. So we don't know how well, or elderly individuals also with their immune response. So yes, they need to be vaccinated, but at this point, we're not sure what the immune responses are in coming up with a vaccine response.
Thank you. So there's a question from Lozelle. The first question is, you indicated after the first dose, I think you answered this question earlier, after the first dose, there's no doubt for the efficacy. So I guess her question is, why do we need to get 2 shots?
Yeah.
And what would explain why is it 3 weeks with Pfizer and 4 weeks with Moderna? I think she got the Moderna and she's getting her dose next week. Congratulations, Lozelle, you're getting your second.
Nice. Yes, right. So first, when clinical trials are being developed or protocols are being written, they put in primary, secondary endpoints, and so the statistical analyses would concentrate on these endpoints. The 95%, 94% efficacy that we see in the Moderna and the Pfizer vaccines were based on analysis after the second dose, not after the first dose. So they did not do any statistical analysis. Maybe they did, but they did not present that. It was powered. The study was powered to actually see a difference between the placebo and the vaccine arm after the second dose. And so the efficacy that we're seeing is based on that, which means if someone stops after the first vaccine dose, we don't know if it's going to be similar to 90%, maybe, or it could be initially, and then suddenly the long-term immunity is not going to be as good. What we have data for, and I think Moderna said that they released that at least the ones that they showed had immunity for 4 months or so. So that duration of immunity, just for now, right? Because it was just administered, so it could be longer than that. But as for now, that's what they say. That will not be based on the first vaccine dose, but the second. The intervals really depend on. So if you look at the phase 1, 2 studies from Sinovac and Sinopharm, you see that there were doses and they looked at intervals, right? 14-day, 21-day, 28-day. So the decision to make it either 3 weeks or 4 weeks depends on the results of those phase 1-2 studies. If they show in those phase 1-2 studies that the vaccine was by 3 weeks was able to really boost neutralizing antibodies and response, and that's what they're going to put into the phase 3 study. Whereas in other studies where they said, oh, maybe it's 4 weeks that you actually see a better immune response, then that's going to move forward for the phase 3 studies. So that's the importance of those phase 1-2 studies.
Thank you, Dr. Macatangay. Another question from Kathy. How can you explain why some people that have been vaccinated can be reinfected with the disease?
I think it's multifactorial. It could be so they may get an infection, but they do not get severe infection. There's still some protection that's there. It may be that the immune response, the neutralizing antibodies weren't enough to do that compared to the amount of antigen that the person was exposed to. These are, or it could be the viral variant. We don't know, right? So right now there's no data to do that. But in these individuals who got the vaccine, so for example, in our institution, we are getting blood from individuals who received the vaccine, and we are several days or weeks after they receive the second dose, and we're trying to look at neutralizing antibodies, T cell responses and whatnot, to see whether, because there's going to be variability in inter-individual variability, and you could actually see this in the side effects, right? Some people have like really robust side effects, really dramatic side effects. Others just got like a pain in their arm, and this may be because of the immune responses that are there. So I think it's multifactorial, and in these cases, I know that people who get reinfected or get infected after getting the vaccine, those are really being studied to find out what are the different factors that are contributing to why this person who received the vaccine got that infection.
Thank you for answering that question. There's a question from Ron, and he's asking about the ABORH. This came out a while ago. People are actually, my patients are asking me what their blood types are. So can you briefly explain how ABORH blood type play a protective part in complications?
You know what, I have to say I haven't really looked into that aspect. I know that people are talking about it, but I have to say I don't know enough about the associations and the pathogen or the mechanistic or the pathogenesis behind that to be able to explain it. I'm sorry.
Yeah, no, I agree. I think there's just some, I'm not very clear about that either, and there's not much data on that. So I don't know how that came about. Maybe I should.
And then I think in the coming days, there are going to be a lot of associations about these different things, and those are some of the stuff that researchers are looking into to see whether there's really a good correlation or association between this and what the underlying mechanism is for these. Or maybe there's just a coincidence or whatnot. So those are the things that are actively being studied. But as of now, I don't know anything.
Whether that's the most common blood type or things like that.
True.
Answer that question. Thank you for that question, Ron. There's a question follow-up from Red Mendoza from Manila Times. He is asking about the temperature. How does temperature affect the efficacy of the vaccines? Why does the mRNA vaccine require minus 80 degrees Celsius freezers? I think while the traditional and inactivated vaccines only need 2 to 4 degrees Celsius freezers. This has been only one of the main issues in the delivery of the vaccines, especially in the Philippines. I guess he's talking about the Pfizer vaccine.
Yeah, so the Pfizer is minus 80, the Moderna is minus 4, right? Or minus 20. Yeah. So
you can do a regular freezer with
regular freezer for the Moderna. This really depends on how it was manufactured, and how it was done. So there's some certain times during the manufacturing that the other things that are included in the vaccine will require really ultra freeze temperatures or minus 80 temperatures in order for it to be stable, whereas in others are not. So this really depends on how the vaccine was manufactured.
Yeah. Thank you. There's another
AstraZeneca also is like minus 20, if I'm not mistaken.
Yeah. So I think that's a big factor in which ones will be distributed in different countries. So so it's probably Pfizer will not likely be a good one to expect in the Philippines for a while.
Or if it's available, it's probably going to be in your big hospitals where they have these freezers.
There's another question from Yvette Chua. Her question is, any vaccine for children in development?
Yes, very good question. Moderna is doing a study in ages 12 to 18. I'm not sure if there are any studies. I think J&J is also looking into some of that. Those studies are ongoing right now, and will be very, very important, especially with schools opening and all of that, to see, because the kids could be carriers of this. They may not have some of the symptoms, but they could be part of it. So um, there is an ongoing study. One I know of particularly is the Moderna study, which is from 12 to 18 years of age. I don't know yet if there are going to be studies for less than 12 years, but I'm pretty sure that all of the different vaccine groups are looking into that.
Thank you for answering that. This is a question from Nicole. Is it advisable for people with severe allergies to get the vaccine? What about cancer survivors? So there's two parts of that question.
Okay, so cancer survivors, well, let's start with allergies. There has been so people who are have severe allergies to food and latex and whatnot, the recommendation of the CDC is still get the vaccine, right? The only thing is the polyethylene glycol, PEG, which is used in certain vaccine drug delivery methods, allowing it to stay there for a longer period of time. That is included in the mRNA vaccine. If you have an allergic reaction to that, then you do not want to get the vaccine because it's involved. But people who have like egg allergies and food allergies and stuff, you are advised, if you are being offered the vaccine, then take the vaccine. There are some studies of some anaphylactic reactions. There doesn't seem to be any correlation at this point that I have read. There may be something there regarding people who have a history of really severe allergies and getting that anaphylactic reaction. I know that there have been some talks whether it is that polyethylene glycol or the PEG that's causing it. But that's currently being studied right now.
Thank you. There's another question from the Philippines. I don't know who asked this question, but any long-term effect, say in 2 to 3 years? I think you answered this question after vaccination.
Yeah, that's the goal, the holy grail of trying to find out whether this is happening. So we don't know. This is the first time the vaccine is being given. So as far as long-term immunity, it's like anyone can. It really depends on a lot of different things. So the viral virus can affect how long do the plasma cells, the T cells, and everything can actually lead to long-term immunity. We don't know for now because the vaccines have just been administered. We hope that it stays there longer. The longest we have right now is the 4 months that Moderna said that how long the immunity lasts. So we're still waiting for additional data to see that. And I think, and this is actually the reason why people are saying that when you get the vaccine, it's like, oh, that's it. We can just hang out and get rid of the mask and just have these. No, we don't know what long-term immunity is. So still, social distancing, masks are still very important, even when the vaccines are available, because we don't know how protective they're going to be or how long that protection is going to be.
Thanks, Beach. We have such an engaged audience. We actually have people from London. We have people from the Philippines. We have from different states. I wish you could just all put where you're calling from. But I think we have a few more questions, so we can probably extend a few more minutes, if that's okay with you.
Sure.
Yeah, thank you. So there's a question about latex allergy and COVID vaccine. Can I have the vaccine if I have latex allergy?
Yes. This is the CDC actually recommends that. For those people who have allergies, if you go Google CDC and COVID-19 vaccine allergies, they actually have a site for all different questions on allergies. And part of that, one of those questions is whether if someone has egg allergy, latex allergy, and all that, those are not included in the vaccine, the mRNA vaccines, at least, that are available. And so the CDC recommends that if you are offered that vaccine, you should take it. But yeah, go look at that CDC COVID-19 allergy. I think they have a whole website just on that, answering those questions on allergy.
Yeah, there's a few more on, for those with food or drug allergies, should they take an allergy medicine before they get the vaccine? So basically, what do they need to do if they're going to
We
get the vaccine and they have.
don't really recommend what's recommended is that if you develop symptoms after, then you can take some of those. So there are certain individuals who have these, severe allergies and whatnot. Initially, so when we got the vaccine, we were allowed to, they observed us for 15 minutes. People who have allergy histories or stuff, they usually observe for 30 minutes. So it's advisable that those people go into vaccine sites that administer the vaccine that have the necessary capabilities to manage people with allergies. So they have epi shots there and EpiPens and antihistamines that are there, things to monitor your blood pressure and all of that would be available. And then when you go there, after you receive the vaccine, you stay there longer, at least 30 minutes to do that. Another important point to remember, if you got an immediate allergic reaction after the first vaccine dose and the mRNA, do not get the second vaccine dose, right? If the reaction that you have is similar to what I mentioned earlier as the adverse events, then it's fine. But if it is a severe allergic reaction, even if it's not as severe, but it was like an immediate allergic reaction after the first vaccine dose, it's not advisable for you to get the second vaccine dose. Now, whether that is going to do for the other vaccine types, we don't know. This is based on the mRNA vaccines that have been approved so far.
So to add on to that, so if I got the the Pfizer vaccine and develop an allergic reaction, can I get the Moderna vaccine? Or even if I didn't get the allergic reaction, can I get Moderna then Pfizer?
Well, so there's no data to do to both of them. Remember, both would have different ways that they were manufactured, which is the reason why in Pfizer it's like 3 weeks and Moderna it's 4 weeks. So there's no data when you combine these two. Although they're both mRNA, would allow us to tell what your immune response or adverse events are going to be. That said, if it is an allergic reaction, it's best not to get the other mRNA because I think they both have polyethylene glycol included in it, and some people might think that would be the one causing the allergy. So,
thank you. There's a question from Anne. She's asking, how about those with Bell's palsy? Is it safe to vaccinate?
Yeah. Yes.
Yeah. So I think she's talking about maybe people who develop Guillain-Barré-like symptoms.
Yes. So that's actually a very important question, and that came up in the AstraZeneca. Remember when AstraZeneca was doing its phase 3 studies, it was stopped for some time because someone had transverse myelitis, right? And then it was approved in the UK immediately, but it took a while for it to get approved here for the phase 3 trial to actually get started in the US because the FDA wanted to make sure that the neurologic side effects is not really immediately and I think the person who got the transverse myelitis actually had a family history of autoimmune disease. That's what they mentioned during that time. That said, in the different trials, specifically so we're involved in the AstraZeneca, there's a specific question there regarding some of these neurologic abnormalities, if you have a history of that, and in the diary. So someone who's participating in these clinical trials, they have this diary where they put in every single symptom that they experience. And this is not just in AstraZeneca, this definitely happened in Pfizer and Moderna, to make sure that the investigators look at all those symptoms and decide whether that symptom is possibly, probably, definitely, or not at all related to the vaccine. That type of data is the one that is submitted to the FDA. Those that cause possible, probable, and definitely secondary to the vaccine is then reviewed to see whether that is acceptable as far as safety.
Wonderful. That's there's a question on, if someone, I think you sort of alluded to this, if someone has been infected with or exposed to COVID-19, can they still get the vaccine? And if so, when would be the best time to get it?
Perfect question. So if you've gotten COVID, can you get the vaccine? Yes, right? Typically, we want to wait around, you could not get it when you are in the acute phase of illness. So if you're sick from COVID-19, you cannot get the vaccine. Wait, and typically the recommendation is 90 days. So if you get COVID-19, you probably will have antibodies to protect you from that. And so they say wait 90 days. If you become exposed to the virus, can you get the vaccine? Well, first of all, there's no data to show that. So this is sort of like a post-exposure prophylaxis, right? If you get the vaccine, does it prevent you from getting a disease when you were exposed? Well, the way to think about it is that when you get exposed, the usual incubation period is around 5 days, whereas the vaccine, it's after the second dose, which is what, 3-4 weeks apart, and it's 14 days after that second dose where you actually see good responses to immune. So it's not going to work. So if you were exposed, if you get the vaccine is not going to be able to protect you, because in 5 days or so, you'll probably get the disease already, where your immune system doesn't have enough antibodies or T cell responses to protect you from getting the disease.
Thank you. So I guess you also answered this earlier. There's a question on how long will the immunity last after I get vaccinated? Will I need to be vaccinated every year?
Yeah, we don't know that. We don't know any data. What we are getting that data, and the investigators are getting that data right now as more and more people are getting vaccinated.
Yeah. Thank you. So from Gigi, is there any disease that is an absolute contraindication for the vaccine?
I think right now the only absolute contraindication is if you have a severe allergy to any of the vaccine components. So if for people who actually have a history of allergies, the idea, if you go to the CDC, you can click on the available vaccines like the Pfizer and the Moderna. They lists there what are the vaccine components, and the idea is for you to bring that to your primary care doctor and look at all those components and see whether you had a history of a reaction to any of those. But it does not have latex. It does not have eggs.
There's a question on pregnancy and fertility. So does the COVID, I think you answered this also earlier when you discussed the mRNA and DNA. Yeah. So does the vaccine cause infertility? And what is the safety of the COVID vaccine in pregnant and breastfeeding women?
So we don't have any data for pregnant and breastfeeding women. So the Moderna, Pfizer, and AstraZeneca actually excluded women who are pregnant and breastfeeding in their studies. So that's why we don't have data. The J&J vaccine that's ongoing does have data. And in fact, one of my friends who was breastfeeding was enrolled in that particular study. That said, the ACOG actually recommends that if you are offered the vaccine, which means you are at risk for getting disease and maybe even getting severe disease, even if you're pregnant or breastfeeding, to take the vaccine. So my sister, who's a cardiologist, she got her first dose when she was pregnant. She got her second dose after she gave birth. She's doing okay so far. She was like, put me in the registry so that people will know more about what happens to pregnant women. And there are a number of pregnant women who actually are being followed to see, so that there's more data on pregnant and breastfeeding women regarding these vaccines.
So basically, we don't have an idea yet whether it should be given to first, second, or third trimester of pregnancy.
No, I don't think there is any data.
Yeah. We have a few more minutes. So can you just comment, I guess this will be the last question. Can you comment on herd immunity?
Yeah, so herd immunity really, so what herd immunity says is if a number of people already have protective antibodies or protected from the disease, then even if the ability of that disease to spread to other individuals is going to be curtailed, or it's going to be protected just because a number of people are already protected in that. Herd immunity really depends per disease, right? So it depends on what type of disease. So the percentage of people that should have gotten vaccinated and the percentage of people who have probably gotten the disease and have protective antibodies, that depends disease per disease. We do not know yet what the herd immunity is for the vaccine, people who receive the vaccine for COVID-19. But that is something that's very much being studied right now. How many people should be vaccinated in a community in order for herd immunity to start? We don't have data right now to show what that is.
Okay, so I mean, there's that one last question, sorry, from Jonah. I got my second dose of Pfizer vaccine on January 19. 14 hours later, I experienced palpitations, chest pain, chest tightness, throat dryness, and I felt I was choking even after drinking water and chills, but no fever. I ended up at the ER for 8 hours. What's your input on this?
Yeah, I mean, some people actually have some severe reactions, and the fact that you got it 14 hours later. So if you look at the data from the New England Journal paper from, is it Pfizer or Moderna, where it says typically you develop the symptoms 15 hours after. So you're within that that period. Immune responses are there. So sometimes you have inflammatory cytokines that are being produced at that time, and people may have that type of reaction, as long as it's not an allergic reaction, which I think that the EV people actually looked at to make sure that you're not getting an allergic reaction to that. Some of these symptoms may be also just secondary to the vaccine. Your immune response may be very robust causing this, or your body's. So.
Great. So thank you. We have such an engaged audience. Unfortunately, we're at the end of our webinar. Let me have our Vice President for the Philippine Medical Association to give our closing remarks. Thank you very much, Bij, for Dr. Macatangay, for joining us today.
Thanks for the
Yeah.
invitation.
Well, thank you. Thank you, everyone. Again, my name is Olan Ona, and I'm the Vice President of PMA New England. And in behalf of our organization, I just want to thank you, Dr. Bij Macatangay, for sharing us your expertise this morning. I learned a lot. I'm sure everyone here learned a lot as well. And if you have any further questions to Dr. Bij, just um, uh, reach out to us. These are our website, our email. We respond to every email that we receive. Again, I would like to thank Dr. Grace Lirio as well for her inputs, and Dr. Candy Buencamino and the officers of PMA who organized this webinar. Last year there were 581 cases, 581 around the world, and 571 is from China, was from China. So there were only 10 outside China. And as what Dr. Bij mentioned, fast forward to 2021, there are almost 100 million cases. So the scientific community worked so hard to create this solution for the pandemic. So we have, for the rest of us, we start praying for a solution. So we have on one hand the science, and the other hand is our prayers, and if you combine them together, and there it is, a miracle. We have the solution. So we encourage you, if you have a chance to take the vaccine, take it. It will help not only ourselves but the rest of us the rest of the world. So anyway, I want to end this. I know we went through beyond the hour. Again, thank you very much for your participation this morning. Dr. Macatangay, thank you very much. Hopefully there's a second one. Okay, so thank you. Have a nice weekend, everyone.
Thanks.
Thank you.